Hello everybody, welcome back to the Mobiochem YouTube channel once more. In this video, we're going to explain an efficient multi-layer approach to model DNA-based nanobiosensors and calculate redox properties in systems based on self-assembled monolayers. So I'm going to start with a brief explanation of the sensing world. A sensor is typically a device that is able to detect and sometimes quantify the presence of an analyte. When the detection task is carried out by an organic or a biological molecule, the sensor is called biosensor. Basically, a typical biosensor accounts for three different components. A bioreceptor, that is responsible for trapping the analyte and create the chemical signal. The transducer, in charge of transforming the chemical signal into another measurable signal. And the signal processing device, which detects or quantifies that signal. There is a huge number of biosensors thanks to the diverse nature of each of their components. In terms of the type of receptor, we can work with an immunosensor, a biosensor based on polymers, or a DNA-based biosensor, among others. In addition, the nature of the signal is also important to design biosensors, and it is another cause of their great diversity. For instance, optical or electrochemical techniques are commonly used in the detection task, although other methodologies are also possible. Today, we're going to focus in a set of biosensors which uses a nucleic acid as a bioreceptor whose measurable signal is electrical. These kind of biosensors are typically named DNA-based biosensors, and the mode of operation of these gadgets is the following. So in a first stage, there will be a charge transfer between the analyte and one of the nucleobases of the nucleic acid molecule. Since the nucleobases are more prone to be oxidized than reduced, a hole is going to be held in the strand. Later on, that hole will be transported along the strand by different suggested processes that will be explained in a future video. Finally, the transport will reach the electrode, which is the signal processing device, and the hole will be transferred from the strand to the metallic surface. In this video, we are going to focus in this last stage, and we are going to develop an efficient approach to study the redox properties of that region. So, basically, this is our original model. However, this model is quite complex to fully study it computationally. Since we are putting the focus on the strand electrode charge transfer, we are only interested in this spatial region of the biosensor. As a result, we simplified the model to a self-assembly monolayer based on a gold surface in which an ensemble of nucleobases are anchored to the metallic substrate by a thiolated linker. Moreover, the system was fully solvated. As stated before, we are interested in the redox properties of the nucleobases that are here. It is well known that the reducer character increases in nucleobases as displayed here. Among them, guanine is the most susceptible upon oxidation, and that's why we chose this moiety to be the target nucleobase in our simplified model. In addition, we also wanted to test the influence of the linker in the redox properties of guanine in a self-assembly monolayer. Consequently, we chose three linkers of different nature to assess that effect. Now let me explain a little bit about the methodology that we have used. We have talked about the redox properties of nucleobases in general. Specifically, we are interested in determining the one-electron oxidation potential, which is a redox potential of an oxidation process. This is justified by the fact that the nucleobases tend to be oxidized rather than reduced. This potential is closely related by this equation to an energetic term that is called the adiabatic ionization energy. Schematically, this is the adiabatic ionization energy. It is the difference in energy between the relaxed cation and the neutral species of a single compound. In order to compute this property, we used the formulation of the Marcus theory, which I explained in a previous video. Basically, we can compute the adiabatic ionization energy using this formula that averages the expectation value of two different energetic terms. The procedure that we conducted is the following. In order to obtain an expectation value, we need to have an ensemble of geometries from which we are able to obtain the average value of the property. Thus, we need to sample the conformational space, and we carried out this task by running classical molecular dynamics simulations. The first energetic term that appears in the formula is the vertical ionization energy, which it is defined as the energy needed to remove an electron from the neutral species, without considering the relaxation of the geometry after the electron detachment. Thus, we ran a classical molecular dynamics simulation to sample the phase space of the neutral species. Afterwards, we selected an ensemble of geometries from that simulation and compute the vertical ionization energy for each of them, obtaining an average value of that property. The second energetic term that appears in the formula is the vertical attachment energy, which is related to the vertical addition of an electron to the cationic species. As a result, we repeated the sampling procedure, but in the phase space of the cation. We chose an ensemble of geometries and calculated the average vertical attachment energy as the mean value of the individual values of the property. Thus, averaging these two terms, we can obtain the adiabatic ionization energy. Notice that this last term is just a correction that accounts for the electron in the gas phase, which has to be included in the equation because it is also included in the value of the reference electrode that we use to calculate the one electron oxidation potential, which is the standard hydrogen electrode. And let me point out another last note. We were interested just in determining the one electron oxidation potential of the nucleobase, so the hole should be placed uniquely in the nucleobase and not in the linker. In order to overcome this problem, we employed the constraint density functional theory, which is basically a DFT with some Lagrange multipliers to restrict the charge or spin in specific regions of the system. With all this information in mind, we are going to discuss the most relevant results and technical details of our innovative approach. Our first obstacle was to obtain the optimal size of the quantum mechanical region applied to the system. It is clear that at least one organic ligand, which means a one-in and a linker, must be included in the QM region. But what about the solvent? What about the gold atoms of the metallic surface? What about the remaining organic ligands of the self-assembly monolayers? As a result, we conducted a convergence analysis for each of those components, and the convergence criterion was the one electron oxidation potential. So let's start with the solvent. The determination of the one electron oxidation potential was conducted for 5 different geometries, each of them represented by a color line. The solid line represents the one electron oxidation potential with an increasing number of water molecules included in the QM region. On the other hand, the dashed lines represent this redox property when using a continuum solvation model, in this case COSMO, to describe the effects of the solvent. From a careful inspection of this plot, it can be seen that convergence is reached when at least 25 water molecules are included in the QM region or when using COSMO. If we plot the mean value of the one electron oxidation potential with the number of water molecules in the QM region from 25 to 60, we obtain these lines. Let's going to remove the plot so that the lines can be observed in an easier way. If we compare these values to those obtained with Cosmo, it is possible to see that the differences in the redox property are relatively small, being comparable. Since 25 water molecules implies a significant increase of the size of the QM region, and there is still other two components to be analyzed, the organic ensemble of molecules and the gold atoms of the surface, we concluded that the best way to model the solvent in this type of systems was the use of implicit solvation model. Let's move on to a similar analysis, this time for the gold surface, in which our aim was to assess how many gold atoms were required to be included in the QM region in order to get a converged value of the 1 electron oxidation potential. So here we have a similar plot to the one displayed for the solvent. Each color line represents the potential against the number of gold atoms included in the QM region. These gold atoms were the closest to the sulfur of the residue included in the QM region by default. From these results, we concluded that 4 atoms were enough to obtain a constant value of the redox property. Last but not least, an identical study was carried out for the organic environment. We included up to 3 ligands in the QM region before convergence was no longer achieved. Again, these ligands were chosen to be the closest to the reference ligand that was included by default in the QM region. We noticed that we needed at least 2 ligands to be included in the QM region. So let's sum up the requirements of the obtention of a converged value of the 1 electron oxidation potential. We needed at least 25 water molecules that we substituted for an implicit description of the solvent to make the QM region efficient. We also needed 4 gold atoms from the gold surface and 2 adjacent organic ligands. Such size of QM region is totally unfeasible to perform a high amount of calculations in terms of the computational time. As a result, an alternative solution was developed in this work. This strategy was named additive scheme, and its basis is as follows. This is our target ligand in water. Now, let's put this molecule together with other identical molecules forming a self-assembled monolayer. This molecule is the only one that is going to be described as a quantum mechanical level of theory with DFT, while the environment is described classically and the solvent through Cosmo. With this description, we are able to compute an approximate value for the 1 electron oxidation potential of the full system. However, if we want to be more accurate by including some quantum effects from the rest of the components of the system, the gold substrate, and the neighbor ligands, we need to include them in the QM region to obtain this term, which is the potential with QM correction. For this, we are going to split the effects of the gold atoms and the organic ligands, assuming that these effects are independent one another and can be computed separately. So, for the quantum effect of the metallic surface, the 1 electron oxidation potential is computed for the system in which 4 gold atoms and the reference ligand are included in the QM region. Then, we subtract the value of the potential when just one ligand is in the QM region to account for the quantum effect of gold in the self-assembled monolayer. Following the same argument, we do a similar thing with the organic environment. We perform the determination of the potential when 2 ligands are in the QM region and subtract the value of the single ligand in the QM region. Both effects are added to the potential of the single ligand in the QM region, and this will be the 1 electron oxidation potential of the self-assembled monolayer with QM correction. In order to assess the validity of our strategy, we compared the potential obtained from the additive scheme to the one obtained from a calculation in which 2 ligands and 4 gold atoms were included in the QM region. This was conducted for 5 random geometries, and here we can see the differences in the final 1 electron oxidation potential from one strategy to another. The error was less than 5% in all the cases. From this assessment, we concluded that the additive scheme was valid to this type of systems. So, once the strategy was validated, we compared the 1 electron oxidation potential for 3 different organic molecules when they were put on a self-assembled monolayer. The structure of these molecules was composed by a guanine bonded to a linker. The difference between the ligands resided only in the nature of the linker: an alkane, an alkene, and an arene. We started computing the potential of each ligand in water as displayed in red in this plot. From these results, we can state that there is not a significant change on the redox properties when the linker is replaced. Then, we computed the potential for the organic molecules in a self-assembled monolayer of identical ligands. Here, these values are represented by blue bars. When each ligand is included in the self-assembled monolayer, we can observe significant changes on the 1 electron oxidation potential in terms of the linker employed. At this point, we added the quantum goal effect represented by green bars. And we did the same with the quantum ligand effect displayed by orange bars, following the additive scheme that was already explained. These two effects were relatively small, and they just slightly modified the 1 electron oxidation potential with QM correction. Here we can see the 1 electron oxidation potential with QM correction, which is displayed in purple bars. If we focus on the red and purple bars, there is a trend that evidences that the reducer character of the ligands increases when the ligand is put on a self-assembled monolayer, no matter which is the nature of the linker. In fact, this trend can be also reported from the calculation of the potential of a single ligand QM region, those with blue bars. This gives us the hypothesis that the electrostatic interactions between ligands of the ensemble lowers the potential, since when the surface is fully described classically, the atomic charge of each gold atom is 0. Thus, these atoms do not contribute to the electrostatic interactions. In order to assess this hypothesis, we performed an analysis to see if the distance of the electrostatic charges to the QM region was the responsible of this decrease of potential, or what is the same, an increase of the reducer character. We took 5 arbitrary geometries of the self-assembled monolayer composed by organic molecules with an alkane linker, and removed every component but the reference ligand. Afterwards, we computed the vertical ionization energy, the vertical attachment energy, and the 1 electron oxidation potential of each geometry of the ligand and averaged their values respectively. Here, the red line accounts for the vertical ionization energy, the blue line represents the vertical attachment energy, and the green line is associated to the 1 electron oxidation potential. Then, we started adding some atoms that were far from the QM region and recomputed these energetic properties. Thus, there was an artificial vacuum sphere surrounding the QM region whose radius was being reduced. If we reduce the vacuum sphere until the full system is taken into account, we obtain this plot displayed in the slide. Here we can see that the vertical attachment energy is affected by the addition of charges near the QM region. This leads to a decrease of the 1 electron oxidation potential following Marcus theory. As a result, we can conclude that the increase of the reducer character is mainly due to the electrostatic effects between the ligands of the self-assembled monolayer. To sum up everything we have talked in this video, we have extracted the minimal characteristics for the computation of the one-electron oxidation potential in terms of the description of the system. The description of the solvent can be carried out by using a continuous solvation model such as COSMO, reducing the size of the QM region. We need to include at least the 4 closest gold atoms to the referent ligand from the metallic surface. And we also need to include the closest ligand to the referent one in the QM region. We have also developed an efficient strategy to compute this potential, splitting the system in its different components and adding its effects to the potential separately. Finally, it has been clear that the addition of a ligand to a self-assembled monolayer modifies its redox potential, and in the case of nucleobases, this fact increases this reducer character. These are the main conclusions that we can extract from this study. So, thanks for watching this video. I want to thank all my colleagues from the Mobile group, also Professor Sergio Díaz Tendero and Juanjo Nogueira, and the computational resources.